Regeneration is to get a little bit further back from what was taken. And right now, of course, it's our language and culture and our history being taught in a school.
Imagine you're in an emergency room. Someone is bleeding out on the table. The doctors don't have time to run tests. They don't know the patient's blood type. Every second counts. And the one thing standing between that person and death is a bag of blood, specifically type O blood. It works for almost everyone. It saves lives every single day. But here's the thing that has haunted scientists for generations. Nobody could fully explain why this blood type exists the way it does, why it spreads the way it does, or why certain people on Earth carry it in such overwhelming numbers. Until now. In 2025, a series of genetic breakthroughs finally cracked open one of biology's oldest cold cases. And what researchers found didn't just answer old questions. It raised entirely new ones about who we are, where we came from, and what our blood has been quietly carrying across thousands of years of human history. Let's start with the basics, because understanding why this discovery matters so much requires understanding what blood types actually are, and why type O has always been the strange one in the group. Your blood type is not just a random label assigned at birth. It is a deeply biological characteristic, encoded in your DNA, shaped by evolution, and tied to your immune system in ways scientists are still working to fully understand. Human blood is classified into 4 main types, A, B, AB, and O. What separates them comes down to something called antigens, tiny molecules that sit on the surface of your red blood cells like identification badges. Type A blood carries A antigens. Type B carries B antigens. Type AB carries both. And type O, it carries neither. That absence sounds simple, but it turns out to be one of the most important biological quirks in all of human history. Because type O blood has no antigens of its own, it doesn't trigger an immune reaction when it's introduced into someone with a different blood type. Your immune system is constantly on guard, looking for anything that doesn't belong, and it uses those antigen markers to tell friend from foe. When type O blood enters your body, your immune system essentially sees nothing unusual. Nothing to attack. Nothing to reject. That's why type O is known as the universal donor. In emergency medicine, it's the blood you reach for when there's no time to ask questions. But the universal donor status, as remarkable as it is actually the least mysterious part of the type O story. Here is what has puzzled researchers for decades. When scientists began mapping blood type distributions across the world's populations, they found something deeply strange about the Americas. Across North America, Central America, and South America, indigenous populations carry type O blood at rates that have no parallel anywhere else on the planet. In some communities, the percentage reaches 90, 95, even close to 100%. The global average for type O hovers around 62%, itself the highest of any blood type, but that number looks modest compared to what researchers found in many Native American groups. A major study conducted in the 1980s in Arizona, examining Native American communities, found that 99% of participants had type O blood. Follow-up research across indigenous communities from Canada down through the Amazon confirmed similarly staggering numbers. This is not normal variation. In biology, when a single trait dominates a population so completely, something significant drove that dominance. Natural selection, isolation, environmental pressure. Something shaped these populations to carry this specific blood type at such extreme concentrations. But for decades, scientists couldn't fully piece together the why. They had theories. They had fragments of evidence. But the full picture kept slipping just out of reach. The leading explanation for much of the modern era pointed to 2 major forces working together: ancient migration and genetic isolation. The prevailing scientific consensus holds that the first humans to arrive in the Americas came from northeastern Asia, specifically from regions around Siberia, somewhere between 15,000 and 25,000 years ago, during the last ice age. At that time, massive amounts of water were locked up in glacier, and sea levels were significantly lower than they are today. This exposed a wide landmass connecting what is now Siberia to what is now Alaska. Scientists call this landmass Beringia. It wasn't a narrow bridge. It was a broad, livable landscape, hundreds of miles wide, with plants, animals, and the resources needed for human survival. Small groups of people crossed this connection over generations, moving into a continent that, as far as we know, had never seen human beings before. They were pioneers in the most absolute sense of the word. And they brought with them the genetic material they carried from their ancestral populations in Asia, including, critically, a high concentration of the type O gene. Once these groups settled into the Americas, something equally important happened. They became isolated. For thousands of years, these communities had almost no contact with the populations of Asia, Europe, or Africa. New people weren't arriving. New genetic material wasn't being introduced. The gene pool that existed in those early founding populations remained largely contained within the Americas. In genetics, this kind of scenario has a name, the founder effect. When a new population is established by a small number of individuals from a larger group, the genetic diversity of that new population is limited to whatever those founders happen to carry. If the founders happen to carry predominantly type O blood, and the evidence strongly suggests they did, then the entire population that descended from them would inherit that same skewed distribution. Combine the founder effect with thousands of years of isolation, and you have a recipe for extreme genetic concentration. Type O didn't just persist in the Americas. It dominated. But isolation alone doesn't fully explain the story. Because over thousands of years of living in the Americas, other blood type genes didn't gradually appear and spread the way you might expect if the distribution were purely a passive result of founding genetics. Type O maintained its dominance actively, across wildly different environments, from the frozen Arctic to the high peaks of the Andes to the dense heat of the Amazon rainforest. That kind of persistence across such varied conditions suggests type O wasn't just preserved by chance. It may have been preserved by advantage. Several theories have been proposed over the years about what specific advantages type O blood might have conferred on early Americans. One of the most discussed involves disease resistance. The antigens carried by type A and type B blood appear to make those blood types more susceptible to certain bacterial and viral infections. The absence of these antigens in type O blood may have made type O individuals slightly more resistant, or at least less vulnerable, to certain illnesses. In a world before modern medicine, where infectious disease was a leading cause of death, even a modest resistance advantage could, over many generations, significantly shift the balance of which blood type survived and reproduced more successfully. Another theory focuses on altitude. Large portions of the indigenous populations of the Americas, particularly in the Andean regions of South America, lived at extremely high elevations where oxygen levels in the atmosphere are significantly lower than at sea level. Surviving and thriving at altitude requires effective oxygen delivery throughout the body. Some researchers propose that type O blood's particular characteristics might have allowed for slightly more efficient oxygen transport under these conditions. If type O individuals had even a marginal edge in oxygen delivery at altitude, that edge could have translated into better survival rates and higher reproductive success in mountain environments. There was also consideration of diet. The early peoples of the Americas were predominantly hunters and gatherers during the period when type O's dominance was being established. Some nutritional research has suggested that type O blood may be better adapted to high-protein, high-fat diets, the kinds of diets that hunter-gatherer communities around the world have historically relied upon. If this connection is real, it would mean that type O blood wasn't just neutral in the context of early American life, it was genuinely well-suited to it. These theories all had something going for them. But they were also incomplete, suggestive rather than conclusive. The mechanisms weren't fully understood. The genetic basis for all of these potential advantages wasn't clearly mapped. Scientists knew something important was happening with type O blood in the Americas. They just couldn't see the whole picture clearly enough. That changed in 2025. A major genetic research effort, involving scientists from multiple institutions and drawing on genomic data from thousands of individuals across diverse populations, published findings that fundamentally advanced our understanding of type O blood at the molecular level. The core of the discovery lay in the ABO gene cluster, the section of human DNA that determines blood type. Scientists had known for a long time the general mechanism by which blood types are determined genetically. But the 2025 research identified previously uncharacterized variations within and around the ABO gene cluster that had not been fully described before. These weren't just minor footnotes. They turned out to be functionally significant, meaning they actually affected how the genes behaved, how the blood type's characteristics expressed themselves in the body, and how those characteristics interacted with the immune system and with disease. Specifically, the researchers found genetic variants that appear to influence how strongly certain immune responses are triggered in relation to blood type. The absence of A and B antigens in type O blood had always been understood to reduce certain immune reactions. But the new data showed the picture was considerably more nuanced. These newly identified variants appeared to modulate the immune system in additional ways that hadn't been previously understood, affecting not just antigen compatibility during transfusions, but also how the immune system responds to a range of infectious agents. This provided a stronger genetic foundation for the long-suspected disease resistance advantages of type O blood. It wasn't just that type O lacked certain antigens and therefore triggered fewer reactions. The genetic architecture of type O blood appeared to actively tune the immune response in specific ways that, under certain environmental conditions, particularly the kinds of infectious disease pressures that early human populations faced, conferred measurable survival advantages. The implications of this finding for understanding indigenous American populations were significant. The research provided a clearer genetic explanation for why natural selection would have favored type O in those populations over thousands of years. The founders who crossed Beringia and settled the Americas carried predominantly type O blood. Once isolated, those populations faced a range of infectious diseases, environmental pressures, and survival challenges. The genetic variants associated with type O blood that the 2025 research identified appear to have given type O individuals advantages in surviving and reproducing under those specific conditions. Over generations, those advantages compounded, and type O became not just common but overwhelmingly dominant. This also shed new light on some of the older data points that had puzzled researchers. The near-total dominance of type O in certain communities wasn't just a passive relic of the founder effect. It was the outcome of thousands of years of natural selection actively reinforcing a trait that genuinely helped people survive. The time capsule of genetic isolation had preserved and amplified something that was biologically valuable, not just present by chance. What makes this discovery particularly rich is that it sits at the intersection of genetics, ancient human migration, and living indigenous communities, and each of those intersections carries its own complexities and significance. On the migration side, the findings add further depth to an already fascinating story about how humans moved across the planet. Lake Baikal, the ancient Siberian lake that sits at the heart of much of the genetic evidence connecting Asian populations to Native American ones, has long been a focus of researchers studying pre-Columbian migration. Archaeological evidence shows human habitation in that region stretching back hundreds of thousands of years. Ancient people along the shores of Lake Baikal were part of complex trade networks that stretched across enormous distances, connecting populations from East and West Asia. The genetic lineages that eventually gave rise to the first Americans appear to have roots in precisely this part of the world. But the migration story has grown considerably more complicated in recent decades. Genetic evidence has revealed that the movement of people into the Americas was not a single, simple event. There appear to have been multiple waves of migration, potentially using multiple routes, not just the Beringia land bridge, but possibly coastal routes by watercraft as well. Some genetic studies have even found evidence of contact between Native American populations and Polynesian peoples approximately 800 years ago, a remarkable finding that suggests pre-Columbian maritime connections across the Pacific Ocean that were not previously understood by mainstream scholarship. The sweet potato, a plant originally domesticated in South America, appeared in Polynesia long before Europeans arrived. Archaeological sites across Polynesian islands have turned up botanical evidence that should not have been there based on conventional historical timelines. The genetic data appears to confirm what the plants had already hinted at. People from the Americas and people from the Pacific Islands met, interacted, and left traces of each other in their DNA. This expanding picture of ancient human connection makes the type O story even more compelling. Type O blood didn't just flow through the veins of isolated populations locked away in the Americas. It moved through people who were, in their own ways and by their own means, participants in a much larger story of human movement and contact across the planet. And today, the descendants of those ancient people carry that history in their blood, quite literally. Modern DNA research has made it possible to trace these lineages with remarkable precision. The genetic markers specific to indigenous American populations can now be identified and traced across generations, connecting living people to the ancient communities whose choices, migrations, and biological characteristics shaped the modern world. For many indigenous communities, this research has a significance that goes well beyond academic curiosity. Generations of colonization, forced displacement, cultural suppression, and the destruction of records and oral traditions left many families disconnected from their histories. DNA testing has, in some cases, helped individuals reconnect with ancestral lineages they had been cut off from. High-profile cases, including DNA confirmation of family connections to historical figures in Native American history, have illustrated both the emotional weight and the real-world significance of this technology for indigenous peoples. But this comes with important ethical dimensions that responsible scientists and commentators are careful to acknowledge. DNA is a powerful tool for understanding ancestry, but it is not the totality of cultural identity. Being part of an indigenous community is about language, tradition, relationship, shared history, and collective belonging, not just genetic markers. When DNA testing is treated as the sole or definitive measure of indigenous identity, it risks reducing rich, living cultures to biological data points, which serves no one and disrespects the communities whose histories are at stake. The responsible use of genetic research in the context of indigenous populations requires meaningful engagement with those communities, respect for their sovereignty over their own biological and cultural information, and a clear-eyed understanding of the difference between ancestral genetics and cultural identity. There are also significant practical medical implications flowing from the 2025 research that deserve serious attention. Understanding the genetic basis of type O blood more precisely has direct applications for blood typing accuracy and transfusion safety. Current blood typing methods are highly effective but not infallible. More detailed genetic knowledge of the variations within type O blood could allow for finer-grained compatibility assessments, reducing the already low but non-zero risk of transfusion reactions in complex medical cases. For patients who require frequent transfusions, people with certain cancers, blood disorders, or chronic conditions, even marginal improvements in compatibility matching can have meaningful clinical benefits. Beyond transfusions, the findings may contribute to personalized medicine more broadly. If the genetic variants associated with type O blood genuinely influence immune function in the ways the 2025 research suggests, then blood type could become a relevant factor in assessing individual patients' susceptibility to certain infections, their likely responses to specific treatments, or their risk profiles for certain conditions. Researchers have noted for years that people with different blood types appear to have different risk profiles for conditions including cardiovascular disease, certain cancers, and infectious diseases. The new genetic data gives scientists better tools to investigate these associations rigorously and to determine which are genuine biological effects and which are statistical coincidences. The persistent challenge of type O blood supply is also worth acknowledging in this context. Precisely because type O is the universal donor, it is always in demand, and always at risk of running short. Hospitals, blood banks, and emergency services around the world face the ongoing challenge of maintaining adequate type O supplies while also meeting routine medical needs. In communities where type O blood is most prevalent, including many indigenous communities, barriers to healthcare access and blood donation infrastructure often mean that the most concentrated source of this critical resource is also the most underserved by the systems that need it. Addressing that gap is a public health priority that the elevated scientific understanding of type O blood should help motivate. Scientists are also pursuing longer horizon solutions to the supply challenge. Research into synthetic blood substitutes has been ongoing for decades, with limited success so far. The challenge is considerable. Blood is extraordinarily complex, performing dozens of functions simultaneously, and replicating that complexity artificially remains beyond current technology. But advances in our understanding of how blood-type genetics interact with immune function could inform new approaches to developing safer and more effective blood substitutes, or to engineering donor blood with enhanced universal compatibility. Gene editing technologies, which have advanced rapidly in recent years, also enter the picture here. It is theoretically possible, using current gene editing tools, to modify donor blood cells to remove the A and B antigens, effectively converting any donated blood into type O. Research along these lines is actively underway. The 2025 findings about the genetic architecture of type O blood will likely inform and accelerate this work, by clarifying exactly what makes type O blood function the way it does and what genetic changes would be necessary to replicate those properties safely in other blood types. Stepping back from the specific findings, what the 2025 type O research really represents is a convergence of multiple streams of scientific progress that have been developing over decades and have now reached a point of genuine synthesis. Advances in genomic sequencing have made it possible to analyze human DNA at a resolution and scale that was simply impossible a generation ago. The price of sequencing a human genome has dropped from billions of dollars to a few hundred, making large-scale population genetic studies feasible in a way they never were before. Machine learning and computational biology tools have made it possible to identify patterns in genomic data that human researchers alone could never have detected. And the accumulation of decades of anthropological, archaeological, and historical research has provided the context necessary to interpret what the genetic data means in human terms. The result is a kind of scientific moment where answers that were previously out of reach are suddenly, almost rapidly, becoming accessible. The mystery of type O blood is one such answer, but it is almost certainly not the last. The same tools and approaches that cracked this particular case are being applied to dozens of other long-standing questions in human biology, evolution, and medical science. The story of where we came from, who we are related to, and how our biology shaped and was shaped by our history is being rewritten in real time, one genome at a time. What the mystery of type O blood ultimately teaches us, when we step back and look at the full sweep of the story, is something profound about the relationship between our biology and our history. The blood flowing through the veins of people in the Andes, in the Amazon, in the Arctic north of Canada. Blood that has been there for thousands of years, persisting through ice ages and migrations and the violent disruptions of colonization, carries within it a record of the choices, environments, struggles, and adaptations of every generation that came before. It carries the crossings of ancient Beringia. It carries the altitude challenges of mountain communities. It carries the disease pressures of a world without medicine. It carries the genetic contributions of those founding populations who stepped into an unknown continent and built civilizations across it. And in 2025, for the first time, science found the molecular language to begin reading that record clearly.